Hello everyone and welcome to Micro-Spectroscopy, enabling research from novel nano-devices to clinical diagnosis, a webinar presented by Photonics Media and sponsored by ANDOR Technology. Before we begin, just a couple of quick technical notes. If you have any problems, the easiest thing to do is to use the question box in the webinar program, and we'll do our best to assist you. We found that if all else fails, simply log out and log back in to rejoin the webinar. I'll introduce our first speaker today in just a moment, but first a few words about us here at Photonics Media. We publish Photonics Spectrum magazine, as well as the magazines Biophotonics, Industrial Photonics, and Europhotonics. Subscriptions are free and forms are available on our website, photonics.com. We also produce a weekly video newscast about photonics called Light Matters that can be accessed on our homepage. During today's webinar, you may type any questions you have in the question box in the webinar program, and we'll answer as many as time allows after all of the presentations are complete. Please be sure to address which presenter you're asking in your question. Our first presenter today is Bjorn M. Reinhard. Bjorn is an associate professor of chemistry and the director of the Nano-Bio-Interface Laboratory of the Photonics Center at Boston University. His research interests include fundamental aspects of photonic and plasmonic nanomaterials, as well as their integration into enabling tools for investigating living systems and cellular processes. We will now begin Bjorn's presentation.
For characterizing plasmonic nanoparticles. Talk about microspectroscopy as an exploratory tool for characterizing plasmonic nanoparticle assemblies. So dark field microscopy is a key technology for characterizing far-field properties of individual nanoparticles, clusters, as well as their assemblies. And in dark field microscopy or spectroscopy, light is incident in the sample plane through a dark field condenser, and this is done to obtain an oblique angle of incidence in the sample plane. In this way, we ensure that only light scattered from the particles in the sample plane, so here, is actually detected by the objective. So after light collection, we can then re-image refocus the light on a CCD camera in case image acquisition is desired. Or we can focus the light on the entrance port of a spectrometer for spectral analysis. So on the right here, you see some typical images of a dark field microscopic setup. In this case, it is based on an inverted microscope. And on the left, you see a spectrometer. Attached to it is a camera. And on the right, you see an enhanced CCD detector, EMCCD, for image acquisition. In the bottom picture, again, you see the spectrometer. This is a side view, the camera, and here the transfer optics, which is available from, for example, ANDOR. And the individual gold nanoparticles of sizes 40 nanometers and above have high enough scattering cross-section so that they can be detected in this fairly simple optical setup. So these green dots are individual nanoparticles. You will also notice that there are some dots which are orange or even red. So these spots belong to nanoparticle clusters because due to electromagnetic coupling between the individual particle, the plasmon, which is a collective electron oscillation within the particle, obtains a redshift of the resonance. And this electromagnetic coupling, which is distance dependent, is used in a broad range of analytical applications. One example is the so-called Plasmon ruler, where we have discrete pairs of noble metal nanoparticles, gold or silver, that are linked by a biopolymer, in most cases DNA. This top picture shows just a typical assembly approach to obtain these devices. In this case, it is based on a template or on a DNA patterned self-assembly. So we have a particle, and we bind two different forms of DNA to this particle, which are complementary to each other. We passivate the particles through assembly of a polyethylene glycol on the surface. Then we hybridize them into a dimer. Afterwards, we can isolate the particles of the dimers of interest through gel electrophoresis, as is shown here. The fastest running band is basically that of the monomers, and then here you see this ladder structure in the reaction mix, and the second band here is the dimer. So now we have access, rational access to these materials. We can isolate these dimers and characterize the structure-function relationship through microspectroscopy that we combine with transmission electron microscopy. So the idea is that you take these dimers, you bind them on the transmission electron microscope grid, TM grid, and then you inspect this grid in the optical microscope. You collect spectra of the object of interest, and after the optical characterization, you transfer to the TEMs, where at higher resolution structural information is obtained. This is illustrated here. So in the top image on the left, you see a dark field image with 1, 2, 3, 4 scattering objects. And on the right, you see the same field of view in the TEM at fairly low magnification. Again, 1, 2, 3, 4, same objects. Clearly, in the TEM, our resolving power is much higher, so we can increase the resolution and now identify the individual objects as dimers or monomers. And we can also obtain very valuable structural information. If we place a analyzer in front of the spectrometer, we can measure we can obtain the polarization results spectra of these individual nanoparticle assemblies. And in the case of the dimer, which is an anisotropic assembly, we can measure the longitudinal mode along the long dimer axis and along the vertical axis. It's shown here. The red one is the longitudinal resonance, and the green one is the vertical resonance. So interestingly, if you now systematically measure the resonance wavelengths of these clusters as a function of separation, this is done here for silver nanoparticle dimers, then you will see that you obtain two characteristic coupling regimes. So there is the electromagnetic coupling regime, where as you decrease the separation, L over D is the ratio of the interparticle separation, the center-to-center separation, divided by the diameter. So as you decrease the separation, your resonance shifts to the red, until at very close separations, you suddenly see a systematic blue shift. This blue shift is currently subject of intense experimental and theoretical investigation. An alternative approach to assemble clusters of metal nanoparticles that are electromagnetically coupled is based on the combination of top-down fabrication and chemical self-assembly. This approach, for example, e-beam lithography, is used to create binding sites in a photoresist matrix that we can then charge positively, and then we can assemble negatively charged nanoparticles on these positively charged surface sites. And after removal of the photoresist, we obtain closely packed clusters at defined location in an array. The nanoparticles used for this approach are usually passivated with some sort of a polyethylene glycol pad that is negatively charged. So here you see the carboxylate head group, which introduce a negative charge to make this charge-guided assembly process on the surface more efficient. And these packs also act as a spacer to control the interparticle separation in the binding sites. With this approach, we can now control nanoparticle cluster sizes at defined locations. This is illustrated here in this SEM micrograph. So by changing the diameter of the binding site created by the e-beam in your pattern, you can now create individual particles or smaller clusters or larger clusters. If you have access to these clusters at defined locations, this opens up the possibility now to create an array of these clusters with sufficient separation between individual clusters that we can read out the spectrum through a micro-spectrometer. And in a second step, after the spectral characterization, we can in principle then again take this sample, transfer it into the scanning electron microscope to obtain structural information. The experimental setup for the optical characterization is the same as the one outlined before. So it's basically a conventional dark field geometry. And the idea is that you now place this individual object, so this individual cluster, in the center of the slit of your entrance of the spectrometer. So this individual dot here, for example, could correspond to an individual cluster. Then you disperse the light through the grating, and that, of course, allows you then to acquire a spectrum of this individual object. And with this technique, you can now systematically investigate, for example, the effect of the cluster size on the resonance wavelengths. This is shown here, for example, where you see individual clusters, dimers, primers, and so forth, and you see the corresponding spectrum. You see overall redshift is increasing cluster size. In addition, you might be interested in identifying morphology-dependent spectral properties. For example, if you think about a trimer, you can certainly think about different configurations. This is shown here. So here we investigate how the spectrum changes from an equally sided trimer, all the side lengths are the same, to a rod of nanoparticles. And this configurational change from the left to the right, this angle here corresponds to the open angle of this angle, leads to a characteristic modulation of the spectral properties, which can be also simulated, and through comparison of the experimental data with the simulation, we obtain significant insight into the underlying electromagnetic coupling mechanisms. The patterning approach is not limited to spherical binding sites. In principle, you can also write anisotropic structures, for example, lines, which then allows you to assemble chains of nanoparticles. This is shown here. We go from one nanoparticle to a dimer, trimer, and so forth. And again, you see a systematic redshift of the resonance wavelengths. And interestingly, as I already indicated before, through control of the pack layer on top of the particles and the assembly conditions, you can change or control the average interparticle spacing in these clusters, which gives you further control over the spectral response of these assemblies. So up to this point, this presentation focused very much on fundamental aspects of plasma coupling in nanoparticle clusters. I will now change a little bit the subject and talk about their potential analytical applications. So in my group, for example, we are interested in characterizing surface receptor distributions on cellular surfaces, and one interesting question could be if these receptors are homogeneously distributed, randomly distributed on the surface, for example in this case here, or if they are clustered, like on the right side here. If they are randomly distributed, you would expect that the average interparticle separation of nanoparticles that are selectively bound to these receptors is larger than in the second case where you have receptor clustering. In this case, the average interparticle separation should be smaller. And this has, due to the distance-dependent electromagnetic coupling, this has direct implications for the recorded spectrum. For large separations, we obtain a more cleaner resonance, whereas for this coupling coupled case, we have a more redshifted resonance. And in these images, these are again dark field images now of cellular surfaces, where we have targeted different surface receptors, CD24 or CD44, with gold nanoparticles, 14 nanometer gold nanoparticles, you indeed see different spots of different color, which are indications of different local differences in the nanoparticle density due to local differences in the underlying receptor density in the cell surface. And this heterogeneity can be confirmed, for example, through scanning electron microscopy. But of course, the advantage of the optical readout is that you can perform this experiment in solution, in principle, even with living cells. A systematic characterization of the resonance wavelengths of the nanoparticles tethered to these receptors on the cellular surface as a function of the average particle density for different clustering levels shows that with increasing clustering levels, so if we go from random, 30% trimers, we observe a systematic change in the resonance wavelengths. Yeah? The larger your clustering level, the more redshifted your resonance is. And this correlation forms the basis for a optical microscopy, which is called plasmon coupling microscopy, which uses optically recorded spectra to infer information about a clustering of receptors on cellular surfaces. So far, this presentation primarily was focused on elastic scattering spectroscopy, but nanoparticle clusters are also of high interest in inelastic scattering microscopies. For example, surface-enhanced Raman spectroscopy is a very interesting analytical tool, and it utilizes that the Raman cross-section can be significantly enhanced if the molecules of interest are bound close to the vicinity of meta-nanoparticles. In particular, if the molecules are located in so-called electromagnetic hotspots, where clusters localize the incident electromagnetic field into very small spatial volumes. This is shown here in these bottom images, where I plotted the E-field intensity enhancement at two different wavelengths. 785 nanometer is a pump wavelength, and that shows this wavelength because this is a convenient wavelength where we have cheap lasers available. And then we have a Raman shifted wavelength at 876.5 nanometer, which corresponds to a 1330 wave number vibrational frequency. And you can clearly see from these plots that the field enhancement coincides at the individual location at these two different wavelengths. And that is very useful because this allows you to enhance the electromagnetic field both at the pump as well as the Raman shifted emission wavelengths. And that is used to boost your Raman signal in surface-enhanced Raman spectroscopy. On the side, I just want to emphasize that although clusters provide already a very strong electromagnetic field enhancement, this field enhancement can be further increased by assembling these nanoparticle clusters into arrays. Yeah? So here you see basically the field enhancement spectrum from the clusters, and due to electromagnetic coupling between the individual clusters, this red curve, a further field enhancement increase is obtained. And I think that's potentially interesting for many applications. So how can we realize the spectroscopy was a typical experimental setup. This is shown here. This time it's illustrated for a upright microscope. So here's an upright microscope. Here you see the spectrometer. And we use a fiber coupled laser, for example, a 785 nanometer laser, and with the choice with correct dichroics and filters, we can now focus the pump wavelengths into the sample plane, and then selectively record the Raman shifted signal on the spectrometer, and then and therefore acquire molecular specific vibrational spectra. There are certainly many applications and examples of this technology. I just want to give one example.
Hey, Bjorn.
Spectroscopy, and allows us to deprotonate this molecule, which generates a Meisenheimer complex in the solution, and as a result of that, we have a very effective mass transfer from the gas phase into the solution. And if we, for example, look at the 1336 wave number band, which corresponds to the NO2 stretch, then we get a very nice signal from these dinitro-toluene molecules. And if we compare that with potential interference, such as diesel, water, pesticides, and fertilizers, all of these molecules give very low background. And so we use this source-based sensor to evaluate what the detection limit for these nitroaromates in the gas phase is in the presence and absence of potential interference, such as water, diesel, and pesticides and fertilizers. And what we see is that in both of these cases, we obtain sub-TPB detection limits with this optical source-based technique. So this is just to emphasize that this inelastic scattering spectroscopy is a very powerful analytical approach and can be used for many different molecules. So with this, I come to the end of my presentation, and I just want to summarize that microspectroscopy is a very useful tool for characterizing the size and morphology-dependent far-field properties of nanoparticle clusters. So it's a very useful tool to fundamentally understand the physics underlying these new electromagnetic materials. And there are clear analytical applications. And one of the analytical applications that I introduced here is the combination of microspectroscopy and nanoparticle cluster arrays to obtain molecular-specific analytical information through surface-enhanced Raman spectroscopy. And I also want to acknowledge that some of this work that I have introduced here was supported by the National Institute of Health. So this, I'm at the end of my talk, and I'm happy to take any questions.
Thank you, Bjorn. We will wait for those questions until we've gone, until after our next two presenters. Our second presenter today will be Ion Nottinger. Ion is an associate professor in the School of Physics and Astronomy at the University of Nottingham and an EPSRC established career fellow. He has established the Biophotonics group at Nottingham, an interdisciplinary research group working at the physical and life sciences interface. His research focuses on optical microscopy and spectroscopy techniques, in particular Raman spectroscopy for studies of biological materials, cells, and tissues. So at this time we'll begin Ion's presentation.
Thanks, Brian, and good afternoon, everybody. So I'm just going to talk today about some work we've done in the School of Physics and Astronomy in Nottingham using Raman spectroscopy, microspectroscopy for imaging cells, and more recent results on cancer diagnosis. So Raman spectroscopy, it's a very versatile tool. So I'm just showing in this slide like an overview of the type of applications we do in Nottingham. So you can see we cover a broad range of dimensions coming down from the nanoscale up to the millimeter and centimeter scale. So for example, Raman spectroscopy can provide you molecular information about the material. And for example, you can combine it with atomic force microscopy, and one could look at the biological nanomaterials by correlating the molecular composition of the biomaterials with, for example, mechanical properties which can be obtained by atomic force microscopy. Or more recent work we've been doing where AFM was used to study the topography and elasticity of cells, and this integrated instrument allowed us at the same time to create label-free molecular imaging of cells. So for example, nucleic acids, DNA, and this image of RNA. Now if we move towards the microscale, Raman spectroscopy is a very powerful tool at looking at cells, live cells, and for that we have developed an instrument which has a microscope with an environmental enclosure. So then one can do measurement on cells, label-free, non-invasive, over extended periods of time, days and even weeks. And here I'm going to give you some example on differentiation of human embryonic cells. We've done a variety of work like time course studies of apoptosis or infection, parasites, and so on. Moving towards the millimeter scale and centimeter scale, we started looking at larger tissues like, for example, dissections from cancer surgery. And here our focus was on overcoming the speed limitations of Raman imaging, which is conventionally done by raster scanning. So we have developed techniques on selective scanning. And these approaches allow you to speed up the measurements and bring diagnosis down to 10-15 minutes for large tissue sections. So now for today what I'm going to focus on is going to be on application of Raman spectroscopy to cells, cell imaging, and I will discuss about differentiation of human embryonic stem cells. And in the second part of the talk I will present some of the results on selective sampling Raman spectroscopy for diagnosis of cancer, and I think I'll focus on just skin cancer for today. Okay, so why we want to look at stem cells? Well, for example, in case of an organ failure, most common type of treatment is transplantation. But we're all aware about the limitations in terms of availability of organs and so on. So stem cell therapy has been like an emerging technology where the aim is to obtain human embryonic stem cells or stem cells from the patient, in vitro, grow them, multiply the number of the cells, produce a sufficient number, and then even differentiate the cells to a specific cell type, phenotype, and then some sort of cell sorting method that will allow us to purify, to select only the cells which are needed for the therapy, and these cells can be implanted in the patient. Equally well, these cells can be used, for example, for testing new drugs or toxic effects of these drugs, or maybe for fundamental studies on the biology of these cells. Now, these also techniques are attractive, and some previous studies have demonstrated that it is possible to obtain, for example, different specific cell types from human embryonic stem cells in vitro, ex vivo, showing here big cardiomyocytes. However, there are major issues, big challenges to overcome before these cells can actually be used in the clinic. So, for example, I'm showing here a figure where we looked at cardiomyocytes derived from human embryonic stem cells, and you can see only the red cell is a cardiomyocyte, the other ones are not cardiomyocytes. So these cell populations are highly heterogeneous. So the question is to how you select these cardiomyocytes to actually use them in the clinic. Because if we use this type of fluorescent markers, they require the cells to be fixed. In other words, they are not live anymore, they cannot be used for therapies. So this is the type of challenge we have been addressing, mainly at this stage where we look at differentiation in vitro. We want to make sure Raman spectroscopy can be used to monitor the differentiation process, to ensure that the cells are differentiating along the right path. And also, again, for cell sorting, it would be nice to have some label-free, non-invasive techniques to select the cells of interest, which then can be used for the therapy. So the instrument, I think I mentioned before, you've got a microscope with an environmental enclosure. Make sure the cells are maintained at 37 degrees, the right level of CO2, contamination is avoided. and then a microscope, a series of lasers, and spectrometers with fast CCDs. So these are typical Raman spectra. You will measure different location of the cell. And then different methods by, for example, doing raster scanning across the cell. You can build spectral images by looking, for example, in this case here is the peak on Raman band which belongs to DNA, and you can see the cell nuclei. And here on the right are the fluorescence images, conventional fluorescence images. This lower spectrum here are from the side which is rich in proteins. And for example, by plotting this peak, you can get images of the proteins. We also cultured human embryonic stem cells on the microscope for long periods of time just to make sure that the cells are happy and you can see the conditions are right for the cells to grow and differentiate. So the first application is finding molecular markers, spectral markers for human embryonic stem cells that have been differentiated to cardiomyocytes. And these studies are carried out on live cells, and we try to identify within a cell culture which cells are cardiomyocytes and which cells are not. And the experiment runs like this. First, human embryonic stem cells are aggregated into what's called embryoid bodies. They are cultured in a specific culture media that will encourage cardiac differentiation. You can see quite a long period of time. Once beating clusters are obtained, they are dissociated and individual cells are cultured in the cell chambers and then placed on the Raman microscope. At the end of the Raman measurements, and the cells will be fixed and then stained with fluorescent markers just to provide us the gold standard, the ground truth for the type of the cells we have measured. So I'm just showing you an example here. Here it's an image of two live cells which have been differentiated from human embryonic stem cells. Recorded Raman spectra, so these are average Raman spectra across the cells. It's been like a pattern raster scan measurement. After the measurement, we stain the cells, we go back on the microscope and we see that only the lower cell is in fact a cardiomyocyte. These two cells here are not cardiomyocytes. So you can see some spectral differences already, mainly in this region here and here. Now we repeat those measurements for tens of cells, and then we do this retrospective fluorescence staining, build a database, and then you can do some statistical analysis. In this case was a principal component analysis, and you can see a very, very clear distinction between the non-cardiomyocytes and the cardiomyocytes. So this provided like a classification accuracy of 95% sensitivity specificity for identifying the cardiomyocytes in a heterogeneous cell population as obtained from the differentiation. Now once we know these markers, for example, one could do more detailed imaging like mapping on the cell, and then we compared with staining for the alpha-actinin, which is a marker from cardiomyocytes, and glycogen. And here we see on the right the Raman map with selected points where some Raman spectra are just shown in this case. And you can see these peaks which are typical to glycogen and these peaks which are typical to proteins, exactly at the location where alpha-actinin is highly expressed in the fluorescence stain. So this shows us that these spectral differences between the cardiomyocytes and other old cells are related to likely to alpha-actinin increase and glycogen. Now, next up, once we understand what the spectral differences are for the endpoint cardiomyocytes, we wanted to go back and to monitor this differentiation process. So what we want to see is we start with embryonic stem cells at this point here, and we want to see how this process takes place by taking Raman measurements along this line. So, for example, here we change the experiment. So we induce the differentiation again of embryoid bodies within 5 days. So a total of about 10, 12 days overall differentiation. But then we did not dissociate the cells, But we placed these embryoid bodies directly on the microscope, and the cells were kept on the microscope over the entire duration of the differentiation. So now I'm showing some, we also done some results to try to figure out at what sort of time points we would expect to see the embryoid bodies differentiating towards cardiomyocytes. So you can see at day 7 of differentiation, already these are fluorescence images on some other parallel cultures. At about day 7, the cells, the embryoid bodies, will be expected to have a high abundance of cardiomyocytes, as these fluorescence images are showing. Again, these images are by fluorescence, and at each stage they will require fixation. So these are not the same embryoid body. So you require different samples for different time points. We also did some flow cytometry to try to quantify what sort of abundance of cardiomyocytes we will expect at the end of the differentiation process. And the results were that at about 12 days, we'll expect about 90% of the cells in the beating embryoid bodies to actually be cardiomyocytes. Okay, so let's see what the Raman results are. So here I'm showing some images taken of these embryoid bodies on the microscope. So day 5, day 6. Now on day 7, we already spotted one of the embryoid bodies starting to beat. You can see in this region here. At day 8, the beating was stronger, indicating more cardiomyocytes. By day 9, the entire body was aggregate was beating. We also did fluorescence staining at the end of this experiment. We fixed the cells, and we see like a strong expression of alpha actinium. So now let's see the Raman spectra, which were taken at each time point. We recorded raster scans over these embryoid bodies to account for heterogeneity, but also to obtain some sort of images of the cells. So what we can see, starting with day 5, when the cells were not beating, you see that these peaks here are not increased. As we go to day 6 and day 7, we can see a strong increase in these bands, which before we saw they were present in the cardiomyocytes. So this day 7 actually is the day when beating was observed, and you can see a major change in peaks. Now, in the case when you have embryoid bodies which fail to beat, that shows that they may not have a cardiomyocytes. Here is it's an image of the fluorescence image at the end of the experiment, and you can see that the peaks remain at the same level. There is no increase in this of the bands in this region, indicating that indeed we didn't have any cardiomyocytes. Now, if we go back and we did this for many embryoid bodies, and just showing here some examples. So on the left are beating embryoid bodies. So you can see with day 5 and day 9. So about day 7, these images show the scores corresponding to the spectral markers which are specific to the cardiomyocytes, which is on this side here. And you can see an increase with differentiation time, indicating an increase in the number of cardiomyocytes in the embryoid bodies, while for the non-beating embryoid bodies, you see no change in these scores. So these studies demonstrate the Raman spectroscopy. It's a very useful tool, non-invasive, label-free, which can be actually used to monitor the differentiation of embryoid bodies towards cardiomyocytes. Right, so I think this is the only application I describe for imaging cells, stem cells. You can check our website for more examples of neuroprogenitor cells and apoptosis and infection. Now I want to change to diagnosis of cancer during surgery by a new technique called selective sampling Raman microscopy. So what is the main problem we're trying to address? So in the case of, so far we looked only at skin and breast cancer, but the problem is for any cancer type in general. So if a patient is diagnosed with non-melanoma skin cancer, which happens on the face, which is what's considered a high-risk area, then most micrographic surgery is the most efficient treatment method. So how this technique works? So first the patient is called in the surgery theater, and then we has a local anesthetic, and then the first layer of skin is removed by the surgeon. Now in this type of surgery, given that we deal with very high risk areas, the surgeon tries to remove the entire tumor, so that will ensure that the risk of the tumor to come back is lower, but at the same time tries to leave in place as much healthy tissue as possible. It's just because to avoid disfigurement, but also to not to affect the function of the skin. Now once this layer of tissue is removed, the patient is sent out of the operating room and the tissue is cut into a thin section, what's called frozen section pathology. Then a surgeon or pathologist will inspect all these sections which have been stained with hematoxylin and eosin. So the aim is then for this pathologist to identify whether there are any more cancer cells in the tissue. If there are no more tumor cells, then the patient is called back for reconstruction and the surgery has finished. However, if there are some tumor cells in this tissue section, then the patient is called in and this process is repeated. So there will be another anesthetic, another layer of tissue removed, and another frozen section histopathology. Typically, most of the patients are cured with 1 or 2 layers. In some cases, more difficult layers, more difficult cases for large tumors, patients can go up to about 4 layers. So what we tried to do is to try to replace this process here with an automated technique, like having a Raman spectrometer or an instrument based on Raman spectroscopy, where the tissue layer can be placed directly with no sectioning, no fixation, and within a few minutes, maybe 10 minutes maximum, to obtain an information like an image which will be able to tell whether the resection margins are clear of tumor cells or not. So then the surgeon could immediately act on this information and remove any tissue layer if it's needed or not. Now what I've described here, most surgery is used at the moment only for non-melanoma skin cancer occurring on the high-risk areas in the face, and unfortunately it's not used for other cancer types. In other cancer like breast cancer or other cancer, you may, the pathology can do diagnosis on selected regions of the tissue, but there is no way that can be done intraoperatively during the surgery or for the entire margin. So there is a very important need for this type of technologies. So we started looking at Raman spectroscopy because we know the Raman spectroscopy has a high diagnosis accuracy based on the very high chemical specificity, and as changes in cells or tissues occur due to the disease, we may be able to pick those up and provide an objective, quantify those changes, and provide an objective diagnosis technique. So the first step for this type of applications is to build what's called the classification model, and that classification model requires a large number of measurements on tissue samples that contain different structures. And then you have and that database will also be based on an annotation process where the Raman spectra obtained from different tissue structures are labeled. So for example, like tumor or epidermis or hair follicle, dermis, and so on. And the idea is then, when a new tissue sample is used for diagnosis, the Raman spectra are run against this classification model, against this database, and then the model will check which class does this new spectrum fit, and with what percentage, what sort of accuracy. So in this way, like a new objective diagnosis can be achieved, and also the level of accuracy can be provided. If this level of accuracy is above a threshold, then it can be used, or if not, then measurement can be repeated or disregarded. So here I'm showing an example where a raster scan on a tissue sample has been measured by Raman, and then a k-means clustering algorithm was used to select the Raman spectra which had similar spectral features. So for example, as you can see this region here, that's tumor. So we can label that as being the spectra belonging to tumor regions, and those would look something more like this pattern here, while this cluster here clearly comes from area of dermis, healthy dermis, and has spectra. So you can see very clear differences. This region here comes from the hair follicles, the epidermis around hair follicles. They're all different. This is just examples. Now, by doing this type of measurements on a large number of recording hundreds of thousands of Raman spectra from 55 patients, here we show the average Raman spectra. You can realize the spectra are very, very similar because you can imagine the level of spectral differences between cancer and the other tissue regions. It's not huge. They are sufficient to provide diagnosis, but they are not that large. So very sensitive statistical methods are needed for the diagnosis. However, what these differences mean, what is the molecular, the main molecules which are responsible for these differences? So for example, if we compare the Raman spectrum of dermis with the Raman spectrum of BCC, of the tumor, this is the difference spectrum. You can see it's very similar to the spectrum of pure collagen. And these are, and that it's very easy to understand. It's just the tumor, which is for example this region here, has very little collagen, while the healthy dermis has lots of collagen. When we compare the tumor, for example, this region spectrum, this region with spectrum from the epidermis, what we can see the different spectrum shows clear peaks which belong to DNA. And this is a spectrum of pure DNA. That's easy to understand as well, that the tumor cells have a higher nucleus to cytoplasm ratio, so have very strong contribution from the DNA. So now as this confidence is built, let's see how yes, so this is like the classification model. This is the receiver operating characteristic for our diagnosis model. So you can see you can achieve very high sensitivity, even 100% sensitivity for a specificity of about 91%. The region, the point on this curve where we like to work is at around 95% sensitivity, which means 95% specificity as well. So all of this is based on independent, of course, validation of the classifiers. Okay, so now let's see how these models work for classifying and producing images for samples obtained from new patients. So here is an example: modular raster microscopy. For cases, these are it's very high correlation, positive correlation for these samples. You can see the tumor here. Here is like the tumor, and the colors here represent the class of the tissue. So that's BCC. The muscles, in this case, there's no muscles. We've got dermis areas, got epidermis in the blue, and so on. So, but the focus was mainly on, so the classifier were optimized for detection of BCC. In this case here, also very good classification. Every small tumor regions is detected. What's also interesting is because the specificity is not 100%. Sometimes we get some false positives around these hair follicles in some cases. Now, for other types of like infiltrating BCC, showing two examples, you can see tumor regions down here are all detected. Again here, they are all detected with the right labels. They are labeled as BCC. Now, for example, in case where we have a tissue which is healthy. We didn't know that. They've got these structures which often are confused with BCC. You can see that the Raman maps tell you straight away that the tissues are clear. They are all healthy. So this is data all from independent patients, and we have some more data in our papers. So I think these results really show that Raman spectroscopy is a very powerful tool for discriminating between tumor and healthy tissue. Now, the problem is that taking this type of raster scan takes a very long time. So if you wanted to obtain, for example, in that case, the size of the tissues were on the level of 2 millimeters by 2 millimeters, or maybe 4 by 4 millimeters, and then the acquisition for those measurements was already in the order of 5 to 8 hours. However, the tissues you obtain in surgery, they can be like a centimeter or 2 centimeters, so that will require several days. So clearly raster scanning is not practical for use during surgery. So what we start working on what's called selective or adaptive sampling. So in other words, instead of using raster scanning, which is a very inefficient method, we wanted to try to identify the main regions of interest in the tissue and then use Raman spectroscopy just to provide the classification, the diagnosis. So how we can achieve this? So we looked at several techniques, and we have got papers published. People who are interested can check. What I'm going to talk today is only about the multimodal approach, where we use the tissue autofluorescence imaging, what's called, to provide a sort of stratified sampling level for the Raman spectroscopy. So this is a technique for this purpose has to have a very high spatial resolution to allow you to obtain high resolution images, to be very fast, the order below like a minute, have very high sensitivity for BCC. So in other words, to be able to identify the regions where one have BCC, but it does not need to have high specificity. So for example, tissue autofluorescence, if excitation is done in the UV, create images like that, where all the bright regions are healthy because fluorescence intensity comes from collagen, while tumor could be any of these dark regions. So for example, in this case, the tumor is in this region here, and you can see they are truly dark. However, that shows very high sensitivity, but however, the specificity is poor because you see here, these regions here, they are which appear dark or gray, they are not BCC. However, this information is sufficient to guide our Raman measurements. I'm just going to give you an example. So for example, by doing for a region of, I think, 4 by 4 millimeters, we can see that by doing the autofluorescence images, we obtain the dark regions, we do some processing, we highlight those regions, then we can do some automated auto-segmentation to break down the image into segments. We throw away the segments which have intensity, collagen intensity higher than a certain threshold, and we end up with these few segments. Now, by selecting only few sampling points for these segments and then acquire the Raman spectra and apply the classification model, one gets this type of image, which tells us that BCC are these regions here. And when we do the raster scanning, what we obtain is exactly the same type of map, but this has taken 40,000 spectra, while the previous sampling was only 350 spectra. It shows the speed up of about 100 times faster by selecting something. So now, by developing this technique, allowed us to look at large tissue samples. So for example, this scale bar here is like 2 millimeter by 2 millimeter. These are full samples. We can see all the tumors are correctly diagnosed. Some other examples with smaller tumors in this case, smaller tumors in this case. And indeed, this case is very, very small tumors, just 2 regions there. You can see correctly classified, only 654 spectra. So typically below 1,000 spectra, which means with current instrument takes about 40 minutes. Now also importantly is to look at tissue samples which don't have, which are clear, they don't have BCC, and you can see within the accuracy of 2-3 wrongly classified segments, this tissue can be identified as clear. Now, importantly, this technique can be applied directly on the tissue layers removed during surgery. So in the example, the confocal fluorescence, all the dark regions are identified, automatic segmentation to identify the interest regions, Raman spectroscopy, classification, and then when we compare with the histopathology, just get the right diagnosis. Another the histopathology. Okay, so this type of measurements again about 20 minutes for full customers. Very quickly in summary, I just showed some examples where Raman spectroscopy can be used as a label-free non-invasive technique. We looked at live cells, showed examples of measuring differentiation of human embryonic stem cells to a cardiac phenotype, and we were able to identify Raman spectral markers and to monitor the intensity of these markers during the differentiation process. And then the second application, I described you some techniques like selective sampling, Raman microscopy, based on autofluorescence imaging that allow us to speed up the measurements of large tissue specimens and obtain diagnosis on the order of 10 minutes to 10-20 minutes for large tissue samples as obtained in surgery. Now, I finished by acknowledging all our collaborators and people who carried out the work I presented today. The collaborators, of course, the funders. And if you would like to learn more about our work, just please go on this website, and we've got more example links to our papers and so on. So with this, I would like to thank you for your attention. Thank you. Bye.
All right, thank you, Ion. Our final presenter today is Gerald Cairns. Gerald is an application scientist for the physical sciences at ANDOR Technology, Belfast, with a particular interest in spectroscopic and time-resolved applications. He has formerly worked in academic research, where his research interests included high-temperature plasma physics, X-rays, thin-film transducers, and plasmonics. At this time, we will begin Gerald's presentation.
Thanks, Brian, for the introduction. Good day to you all. I will give a brief overview of the components of a modular system and relate what ANDOR is providing in this regards. The basic idea is that we can take individual self-standing components, such as a microscope and spectrograph, and integrate these into one functioning system. The two key additions that we need to enable this integration are the necessary mechanical and optical accessories and the software for the control of the whole system. Here are some examples with inverted microscopes. On the left-hand side, we have direct coupling using C-mount adapters between the spectrograph and the microscope. On the top right-hand side, we have a cage system for coupling, and on the bottom right-hand side, we've got an enclosed relay optic, and this may be useful in situations where there are space constraints. Configurations based on upright scopes are relatively easy to set up as well. Here are two examples, one with a cage system on the left, and on the right-hand side, we have direct coupling which incorporates a turning mirror. Now, in the example configurations I've highlighted so far, both imaging and spectroscopy can be carried out on the same camera, on the spectrograph. There are other configurations where people use optical fibers connected to the access port of the microscope and which deliver the spectral signal to the spectrograph. In these cases, imaging cannot be carried out with the same camera. Key to creating a fully integrated system is having the software to control and synchronize all the components. Recently, ANDOR has developed plug-in modules for the popular free and widely available Micromanager software to incorporate control of the Shamrock SR193i spectrograph. Shown here are the data windows of Micromanager for data entry, for multi-dimensional acquisition setup, and for the display of the spectra as they are required. Many users develop their own customer applications using the likes of LabVIEW, MATLAB, and so on, along with the relevant SDKs for the system components. A summary of the main components required to configure a system are shown here, with the microscope and sample at the heart of it. Spectrographs with good image correction are generally needed where direct coupling is concerned. ANDOR has developed SR193I with such characteristics in mind, that is, excellent image quality with high throughput. CCD cameras are the detector of choice with both imaging and spectroscopy sensors being used. Two technologies which are having significant impact on microspectroscopy measurements are EMCCD and LDC technology. These provide higher sensitivity detection. This can be critical in experiments where, for example, the intrinsic signal is extremely low, or one requires the fastest rate of acquisition. That is, they're required to use the shortest exposure times possible, or in situations where one wants to preserve the lifetime of the sample with the minimal excitation energies. The Newton EM and Axon Ultra are often chosen in such circumstances. A range of accessories, both mechanical and optical, are needed to complete the system. These include wide aperture slits, cage systems, filters, and so on, and are readily available from quite a number of different suppliers. Light sources are a necessary part of the system for the excitation and illumination of the samples. As already mentioned, the software to control the multidimensional data acquisitions of the system is the final ingredient. As part of the ongoing custom developed solutions, plugins are now available in Micromanager to add spectroscopy capability to it. Thank you for listening, and if you have any questions or would like more information, please feel free to contact us.
All right, thank you, Gerald. And thank you to all of our presenters today. Unfortunately, Eon could not be with us for this Q&A session, but Gerald and Bjorn are with us live to answer any questions you may have. So at this time, I'd ask you to enter any questions you have in the question box of the webinar program. We are 4 minutes past the hour now, but we'll get to as many as we can within the next couple minutes. And any questions we don't get to will be posted on our website along with the archived version of this webinar, photonics.com/webinars. And when asking your question, please address which presenter you are asking. Thanks. So we'll go ahead and get started here. Bjorn and Gerald, are you with us?
Yes.
I'm here, yeah.
Okay. All right, our first question, let's see here. All right, what typical spatial resolution is achievable on the system using a 60 times objective?
The question for me, or?
Yeah, I believe so.
Okay. So I mean, these optical setups that I have introduced in my presentation, those are diffraction limited, right? So so basically with a 60x objective, you know, the typical numerical apertures, that we can expect for these objectives. We can typically, in the green wavelengths, expect a resolution of something like 500 nanometer. So that's basically, I mean, in those applications where I talked about localization, individual, particles, for example, That's the resolutions which we can localize them. But utilizing these tricks provided, for example, with the electromagnetic coupling between the individual particles, whose distance information is encoded in the frequency, so in the wavelengths, we can probe distances below that diffraction limit.
Okay. Our next question is for Gerald. Sirkin is asking if you can elaborate on the difference between the relay optics and direct coupling as shown in Dr. Reinhardt's setup.
Well, the relay optics will typically incorporate lenses or in some situations mirrors. And the idea is basically to facilitate the placing of the spectrograph at some distance from the microscope. So there are situations where there are spatial constraints, or as some of the examples in the presentations here, where you've got incubator housings taking up a lot of space around the microscope and sample. So in order to get the spectral signal to the spectrograph has to be located further away from the side port. So that's where the benefit of the relay optic comes in. Obviously the advantage of direct coupling, where that is possible, is that you have less optics within the system, less losses, and less optics to worry about in terms of alignment.
All right, our next question is for Bjorn. Ahmet is asking if you could explain again how do you get microspectroscopy of arrays? Is it hyperspectroscopy? He's wondering.
In principle, I mean, you could do hyperspectral spectroscopy, on these samples. I'm not sure exactly. So I guess the question is, if it depends on the separation between the individual particles in your array. So if the separation between the individual particles are above the diffraction limit, then basically we can read out each individual particle, one at a time, and we can basically obtain spectral information from the individual particles. If the particles are closed below the diffraction limit, then basically, I mean, they're diffractively coupled, and the signal that we obtain is basically, a diffraction signal that that is a consequence of the interaction between multiple particles in the array. So it depends really on the morphology of the array, what the signal is that you detect, and I mean, there are applications for both of these cases.
Okay, our next question is for Gerald. This is from Zhang Chao, wondering what is the function of the U manager?
Micromanager.
Oh yes, yes.
Okay, so Micromanager is a software that's out there used by microscopists quite widely, and it's popular software and freeware. So what has been developed are plugins to allow spectroscopy to be actually carried out using it. So these plugins allow you now to control the spectrograph, control the setting up of the spectrograph, and then to control multi-dimensional acquisition. So if you wanted to configure your microspectroscopy system to do two-dimensional Raman mapping, for example, a 2D scan, then that can be handled by the Micromanager now. So it's added functionality to that widely available software package.
Okay, this is going to be our last question for today, but it's for Bjorn. Pablo is asking, when you measure plasmonic features, you measure transmission or reflection? In the plots you showed, what is the normalized intensity?
Right. So in these measurements that I have shown, it was not absorption nor transmission; it was scattering. Yeah? And the scattering, the intensity that was shown in these pictures. I mean, we ensure that we look at scattering due to the oblique incidence of the light, so that basically only light that is scattered from objects in the sample plane is detected. And so the intensity, you could
with a careful analysis of the intensity of the incident light, you can get absolute scattering cross-section from that, but the data that I have shown are basically just relative intensities. So they're basically just normalized.
Okay, thank you. That's all the time we have for questions today. In the next few days, we'll work to get you the rest of the questions answered and posted to our webinar page, which will also include a link to the archived broadcast. If you missed any part of today's presentation or want to share it with your colleagues, you'll be able to find the recording tomorrow on photonics.com/webinars, where you can also find our upcoming webinars and over 40 past presentations. So once again, thank you for attending today's webinar presented by Photonics Media and sponsored by ANDOR Technology.